Emerald cockroach wasp, Ampulex compressa. The emerald cockroach wasp, scientifically known as Ampulex compressa, is a solitary wasp belonging to the family Ampulicidae. It is renowned for its extraordinary reproductive behavior, which involves stinging a cockroach and utilizing it as a host for its offspring. This intriguing parasitic wasp's natural history showcases a remarkable example of nature's complexity. Emerald wasps primarily inhabits tropical regions across Africa, South Asia, Southeast Asia, and various Pacific islands. These flying wasps are most abundant during the warm seasons of the year when their cockroach prey is prevalent. Notably, an attempt was made to introduce Ampulex compressa to Hawaii in 1941 as a biocontrol measure. However, this endeavor proved unsuccessful due to the territorial tendencies of the wasp and the small-scale nature of their hunting. The species is not confined to Southeast Asia alone, it has also been found in the Brazilian states of São Paulo and Rio de Janeiro. It is believed that Ampulex compressa may have entered Brazil through the ports of Santos and Rio de Janeiro. This wasp features a metallic blue-green body, with the thighs of the second and third pair of legs highlighted in red. The female, which is the larger of the two sexes, measures around 22 mm in length, while the male is notably smaller and lacks a stinger. Male size can be influenced by emerging from a smaller or superparasitized host. The species undergoes 4 larval stages during its development. In these stages, the initial, younger larvae are observed as external hemolymph feeders on the paralyzed cockroach's legs, while the final and star larvae feed internally. The wasp pupates within a thick, spindle-shaped cocoon, often found inside the deceased cockroach within its underground burrow. Female wasps are known to sting a specific species of cockroach, such as Periplaneta americana, Periplaneta australasia, or Naphoida rhombifolia, twice. These stings deliver venom that serves to manipulate the behavior of the cockroach. The initial sting targets the cockroach's thoracic ganglion, causing a temporary and reversible paralysis of its front legs. This biochemically induced paralysis facilitates the second venomous sting, which is directed precisely at the cockroach's head ganglia, affecting the section responsible for controlling the escape reflex. The consequences of the second sting are remarkable. The roach will first groom extensively and subsequently become sluggish, with a notable lack of normal escape responses. The venom has been reported to block receptors for the neurotransmitter octopamine, which is essential for the roach's normal behavior. With its victim incapacitated, the wasp begins a gruesome process. It chews off half of each of the cockroach's antennae and feeds on the exuding hemolymph. Subsequently, the wasp, too small to carry the roach, leads the victim to its underground burrow, effectively pulling one of the roach's antennae like a leash. Inside the burrow, the wasp lays 1 or 2 white eggs, each about 2 mm in length, between the legs of the roach. After this, it proceeds to fill the burrow entrance with surrounding debris, serving both to keep out predators and competitors and to keep the roach within. The roach, now devoid of its escape reflex, remains in the burrow as the wasp's egg hatches approximately 3 days later. The hatched larva lives and feeds on the roach for 4 to 5 days, both externally and internally. Eventually, the larva chews its way into the roach's abdomen and lives as an endoparasitoid, consuming the roach's internal organs over a period of 8 days. This process eventually results in the death of the roach. The fully developed wasp emerges from the roach's body, leaving behind a pupal cocoon. The entire development cycle is expedited during warmer seasons. Mating is a brief process, taking about a minute, and a single mating session is sufficient for a female wasp to successfully parasitize several dozen roaches. Unique aspects of predation. Ampulex compressa stands out among predators due to its unique approach. Unlike many venomous animals that immobilize their prey entirely, this wasp initially leaves the roach mobile but significantly modifies its behavior. While a stung roach exhibits a sharp reduction in survival instincts such as swimming and responding to pain for about 72 hours, its motor abilities, such as flight and the ability to flip over, remain unimpaired. The manipulation of the cockroach's behavior begins with the wasp's delivery of 2 precise stings. The first sting targets the prothoracic ganglion, leading to a 2 to 3 minute paralysis of the front legs. The venom of this sting includes significant quantities of gamma-aminobutyric acid, GABA, and complementary agonists taurine and beta-alanine. Together, these substances temporarily block motor action potentials in the prothoracic ganglion by enhancing chloride conductance across nerve synapses. The second sting, delivered to the subesophageal ganglion, SEG, is more precise and significantly longer. The wasp actively searches for the SEG during this sting, The second sting inhibits the cockroach's ability to walk spontaneously but allows it to right itself and swim, though not to run. It also induces excessive grooming and metabolic alterations in the cockroach, preserving nutrients for the wasp larva. Researchers have simulated this state of altered behavior by injecting procaine into the SEG, which led to reduced neuronal activity. The venom's disruption of octopaminergic modulation in the roach's ganglion limits the effectiveness of octopamine, the neurotransmitter controlling muscle contraction during sudden movements. Cockroaches have also learned to defend themselves. They either kick the wasp away as it approaches, or shakes violently till the wasp gets thrown out. These acts of self-defense increases the survival of the roach by 70%. In conclusion, Ampulex compressa, the emerald cockroach wasp, is a marvel of nature with its intricate reproductive behavior and parasitic lifestyle. Its unique method of manipulating the behavior of its cockroach prey, allowing it to serve as a living host for its offspring, is a testament to the astonishing adaptations found in the natural world. This parasitic wasp not only sheds light on the complexities of predator-prey relationships but also raises intriguing questions about the ethics of studying such remarkable phenomena.